So good evening and welcome everyone to our second episode of PRISM Talks. For those who are joining us for the first time, the PRISM Talks, which stands for Progress in Research, Innovation, Science and Medicine, is a talk show which has been, or a webinar which has been organized by Chennai Liver Foundation from Chennai, obviously. So we intend to do this once every month on various topics which is of interest to everyone. Basically, and what we are trying to do is bring in some very high-end speakers to your offices, to your rooms, to your laptops, to so that to encourage you to do more work, to interact with such stalwarts, so that we can do more research, we can do more collaborative work. In that aspect, today we have a second talk, and the second talk, the speaker who is already online, is a very good friend of ours whom we've known for a very long time, is Dr. N. Kumarasamy. Sir, do you want to say hello so that I can then introduce you?
Yeah. Hi everyone.
So thank you, sir. And so a brief introduction of Dr. Kumarasamy. He is the Chief and the Director of Infectious Diseases Medical Center at the Voluntary Health Services Hospital in Chennai, and directs the clinic which provides medical care for persons with HIV, viral hepatitis, tuberculosis, COVID, and other infectious diseases in Chennai, southern India. For those who are working in this field, would definitely know Dr. Kumarasamy. Dr. Kumarasamy is the Director of the Chennai Antiviral Research and Treatment Clinical Research Site, or it's called CART in short, which conducts therapeutics and biomedical prevention clinical trials in HIV, viral hepatitis, and other infectious diseases. He's involved as a clinical investigator for several ongoing clinical research projects with the University of California, Brown University in Providence, Harvard University, University of Colorado, University of Washington, University of New South Wales, Sydney, Australia, for the Treat Asia Network, King's College London, the National Institute of Health in US, and of course the WHO and the Indian Council for Medical Research. He's involved as an investigator for clinical development in several antivirals. Dr. Kumarasamy is a clinician and a researcher in infectious diseases with more than 25 years of experience, and has published over 475 manuscripts in peer-reviewed and medical journals. He's conducted several landmark antiretroviral clinical trials, which has impacted treatment guidelines. His publications on impact of antiretroviral therapy has led to several antiretroviral scale-up programs in developing countries. He's an advisor and expert panel member for the WHO ARV Treatment Guidelines Committee, and a clinical advisor to CHAI, or the Clinton Health Access Initiative. He has a particular interest in long-term impact of antiretroviral therapy in resource-limited settings. He serves as an editorial committee member for several medical journals. And it goes on and on. I think we have to stop here. And what I would do is we can share a link of all his publications in the chat box very shortly. So before he goes on, one more housekeeping is all your mics will be muted by default. And if you have a question, please do raise your hands, and please do drop a message to us or in the chat box so that I can ask a question to Dr. Kumarasamy, and that's how it's going to work. We also will provide a link at the end, which where you have to go register, put your names and details, so that we can send you an e-certificate. That will be done about 10 minutes from the close of the program. So without much delay, I'm going to invite Dr. Kumarasamy to give his lecture. Thank you, sir.
Thank you, Dr. Vivekant, and thank you, Chennai Liver Foundation, for inviting me, to do this PRISM talk. So what I'm intending to do, today is I'm just going to share the slides. Can you see my slides?
Yes, sir.
Dr. Vivek?
Yes, sir.
And it's moving, right? Okay.
Yeah,
right. So what I'm intending to do today is I'm going to talk to you on what's the impact of clinical research on changing treatment guidelines. This is mainly a research webinar. Many of you know about variety of type of research. You may wonder, why we do research. and what you're going to do with this research, and the data you generated, what others are going to do this. Is it just like a presentation in a conference or in a publication, or put it in your, in your biodata stating that you got published so many papers, or what impact? So what I intend to do in the next 30 to 40 minutes is just walk you through, by coding some of the research we carried out at our research center at Voluntary Health Services at our Infectious Disease Medical Center, and how it had impacted the guidelines. Before I go into that, I'll just briefly walk you through some of those, terminologies in different trials. Starting from, very simple terminology, like what is clinical research? Here, many of you might be a well-fledged researchers, you might be a repetition, some of them might be a beginners. So clinical research is a branch of healthcare science that determines safety and effectiveness, also the efficacy of medications, not only just medicines, but also devices and diagnostic products and various treatment regimens intended for human research. Research is done in variety of aspects. When you talk about clinical research, it is mainly for human use. And this may be used for prevention of a disease, treatment for a disease, or for diagnosis, or for relieving certain symptoms of disease, particularly in the cancer, especially on the palliative treatment. And for listeners here, it's very important, clinical research is very different from clinical practice. So in clinical practice, established treatments are used. We prescribe various medication for any illness, which are already been well established and part of guidelines, you use this. So you should not mix clinical research and clinical practice. While in clinical research, we collect the evidence, by doing the research to establish a treatment, which in future will be validated by a variety of different bodies, which you'll see that in my presentations. Then it'll come into the clinical practice. So, and clinical research is very different from, clinical practice. Again, there are different types of clinical research. There are different ways people classify. For this audience, and I like to classify in a way that, just a little lower this screen. Sorry, I think my slides went somewhere. Yeah, I got it. So, sorry about that. Yeah. So you have different types of Vivek?
Sir,
I think my screen. Yeah, my screen has been paused by your.
No, sir. We can't do that, sir. You're sharing your screen. We can't pause your screens. Can you go back on your slide?
It's not moving for some reason. Because I don't have a control over that.
Can you?
I can. I'm sorry about the audience. I can just stop it and again get into this. Yeah, now it moves. Right. So variety of type of clinical research we have. So for this audience, I'm going to classify this as in a very simple way, which will be pertinent to my talk, is a treatment research, which is generally involves the intervention of a medication or a behavioral therapy called psychotherapy, or new devices, or new approaches to any surgeries or a radiation therapy. So we also have what is called the prevention research, which looks for better ways to prevent diseases or disorders from developing or returning. And different kinds of prevention researchers can be using certain medications, using vaccines, you can using certain supplements like vitamins and minerals, or even doing simple lifestyle changes. You can do prevention research, how various people are now carrying out using exercises and yoga, so on and so forth. And diagnostic research refers to the practice of looking at for better ways to identify a particular disorder or identify a disorder or a condition. Also, we do have epidemiological studies which seek to identify patterns of and causes and control of disorders in a group of people, whether any particular disease is being present. we have screening research, which aims to find the best ways to detect certain disorders or health conditions. We did a lot for a variety of things. We did with COVID, like we wanted to screen out so many people who got a COVID in particular, presenting to a hospital or a region. So these are all called screening research. You also have quality of life research. Don't think all the time so clinical trials and clinical research is pertaining only to medications and devices. Also, think certain tools, behavioral, but it's to quality of it's particularly been carried out in patients who have got any chronic illness, like diabetes or malignancy or HIV or any chronic diseases. And of late, one of those star attraction in clinical research is on genetic studies. This again aim to improve the prediction of a disorder by identifying or understanding how genes and illnesses may be related. This is mainly to develop various tailor-made treatments based on patient genetic makeups. Today, with advanced science, one can look into the whole genomic makeup and can map what type of diseases they can develop in the next 10 years, and based on that, what prevention they can be make. Also, there are various gene treatment. It's also been tried, especially by introducing certain molecular scissors and splicing the genes. But these are some of the things, still in the primitive stages, which something probably will take up a long leap, over a period of years. So moving to the clinical trials, which I'm going to talk about a lot today, we got different phases of trials. We don't do a trial just like on a patient before testing on different other mechanism how it has been safe. The first phases in clinical trial is called phase 1. What the scientists do is they experiment a drug or a treatment in a small group of people for the first time. It has been developed. The researchers evaluate the treatment's safety and determine whether it is safe, and also try to find a safe dose range and identify any early side effects. This is what is called phase 1 trial. Usually it has been conducted in a very small number of people, mostly in healthy volunteers. People have got no disease and perfectly fine, how it has been safer. And also, it's also been done in diseased people, just to find out the safety in a diseased individual. Safer in healthy volunteers is different from safety in people who are already been deceased. This is carried out in a small number of people, anything from 20 to 100 individuals. The next step when it passes the phase 1 is called phase 2. This is again the experimental drug or treatment. We have very large people to see whether it is really effective, whether the drug is really working, again an infectious agent or non-communicable diseases like diabetes, hypertension, or anything, or malignancy. And also to further evaluate the safety on a little longer duration and also on a larger number of individuals. Usually the phase 2 includes anything from 100 to 300 individuals, which is something required to do a clinical trial to get a good statistical significance. And when it passes the phase 2, then it goes to a phase 3 trial, where again, this is an experimental study or a treatment given to a very large number of group of people, mainly to assess and confirm its effectiveness and monitor the side effects. We have been seeing from phase 1, phase 2, now in phase 3, and compare this with a commonly used treatment. Always in phase 3, we compare the experimental drug against what is currently being used in clinical practice, what we call standard of care. So what we use as part of our prescription, and collect information that will allow the experimental drug whether it can be used safely. Phase 3 trials are usually done on a very large number of people. Again, based on a sample size calculation, which I'm not going to talk today on that. So it could be roughly anything from 1,000 to 3,000 study participants to assess its efficacy on phase 3. Not everybody who have been doing clinical trials and phase 2 and develop a molecule will go to phase 2 or a phase 3. Based on different literature searches, it's been found that only around 25 to 30% of the drugs which are being developed, which has gone through phase 1 and phase 2, had entered or advanced to the phase 3. Remaining all those 70% of the medications have been killed, because either they are lack of efficacy or they are being very highly being toxic, they are not moved forward. So that means not every medicine which are being tested in phase 1, phase 2 will move forward towards being phase 3. Once phase 3 is being established, then it go to the regulators and various regulations, which again, there might be another presentation another day for you, especially on how that regulations happens. Then it's been approved by the regulator, become part of the guidelines. Once again, it's become the part of the guideline, then it become part of a clinical practice. You prescribe this medicine for any certain indication, indicated for diabetes, hypertension, malignancy, HIV, hepatitis B, or anything on. So once again being prescribed, also we do a study which is called phase 4 trial. This is what is called post-marketing study, which have already been approved, prescribed for the individuals, again with a set of questionnaires. These prescribing physicians collect this information mainly to assess the long-term effect of this drug, and as well as any adverse effects patients have been developing while they have been part of the routine clinical practice, how is its benefits on its on a long run, how it's best used. These are what are called phase 4s. This will also be an advice these manufacturers whether if there are any errors in this development to fine-tune, or whether you need much more better efficacious drugs or better tolerable drugs or not. So these are the various phases of clinical trials. With all this background, I'm going to make a short talk on impact of the clinical trial. I'm basically an infectious disease physician. I'm the chief and director of the Infectious Disease Medical Center at Voluntary Health Services Hospital, VHS, here in Chennai. So the mission and vision of our clinical unit is to provide scientific, ethical, and affordable care for persons with HIV, viral hepatitis, and all the other infectious diseases, particularly on viral diseases. And also to carry out clinical and prevention research in viral and other emerging infectious diseases, and to provide technical support for guideline makers, mainly to update the treatment guideline, national guidelines, WHO guideline, other international guidelines, and also to train healthcare workers in infectious diseases, and also provide technical assistance to various national, international organizations, especially to develop public policy in the field of infectious diseases. And this is something what been involved, and the facilities what we have in our center. We also do a variety of research to advance clinical and prevention therapeutics, in mainly doing clinical research in collaborations with US NIH, with AMFOR, World Health Organization, ICMR, DBT, and with various academic universities across the globe, in the North America, in Europe, in Australia, in Asia, as well as in UK. All this research done have led to more than 475 peer-reviewed manuscripts, which have changed many treatment guidelines in the field of infectious disease, particularly HIV, viral hepatitis, TB, COVID, and many other emerging infectious diseases. With that experience of conducting these clinical trials and changing these guidelines, I'm going to walk you through to why we should do clinical research and trials, how it had impacted treatment guidelines. I picked up one disease, HIV, because that's one thing we have mastered, and we keep doing a lot of trials and change policy. So this is something what these epidemiologists are estimated that how many people are living with HIV globally, around 39 million people. And there are still new infections being growing. There's a data from 2024, around 1.3 million people. And particularly in our region of Asia Pacific alone, 23% of all new infections globally is from our own region. So that means this HIV disease itself is being a real cause of concern. Again, when you have been doing research, you can do research in a variety of diseases. So I'll always suggest to do something which are very critical. Now, which will really be causing major epidemic, pandemic, which is really have unanswered questions. So which will again quickly will contribute to a lot of literature which is being needed. Today, HIV is something still being spreading. There are new infections coming up despite so many other therapies. Again, in India, our national program have estimated around 2 to 2.5 million people living with HIV. But wonderfully, we have seen there's a huge decline in new infections and death rate. This again, I'll attribute many of the research, what many of us done, which is the science has contributed to this, which you'll hear from me now from the next slide onwards. So in HIV, what it does is usually the minute when someone acquires the disease, it goes and destroys the white blood cell, T cells. As a result, they lose their immune system. They develop a variety of infections around them. They can develop TB, the fungal infections, viral diseases, and so many things, so on and so forth. And these individuals die of those infections because of these viral disease causing immune destruction. Before the 1995-96 era, we never had any literature from India or any from developing countries, anything about HIV disease, how it presents and how it causes the disease and anything. We always used to go through the Western textbooks, particularly Harrison textbook on medicine, which is like a golden Bible to many medicine specialists. So what's been described is the common illness in the US and many developed world is a pneumonia called pneumocystis pneumonia, a malignancy called Kaposi's, and Mycobacterium avium infection, most common problem. So this used to be the description, those years, before the 1995, because we never had any literature. When we started seeing our patients, we always looked for this. We didn't see this as the most common thing. Now, to a present, PCP has not been common. That made us those years, almost like 30 years back, hey, this disease is something different in India. I think we need to do our own research. That's a time at our center here in VHS, we started a very simple research. Who are patients who have come, we started collecting their clinical data, that is clinical observational data, in a simple Excel sheet. At that time, we didn't have any sophisticated software, single Excel sheet, collected the information, their demographic, how they presented to your clinic, what's the presenting infections, opportunistic infections, what are those various co-infections, what's the treatment that we prescribe them with their medications, and if they develop any simple side effect, because adverse effects, what we described in the US was very different, from our patients. With those, the dates, we captured all those. So OCS in a very simple Excel sheet we developed. Now we have a very sophisticated database, the one which I've been showing, all those, even last seen date, if they die at those years, we never had good treatment for HIV disease. People, die when they have an AIDS-defining illness. But now we have better therapies available, reason for death, all those being captured. We did the first 100 patients in analysis. We have never existed in Indian literature or in a developing country. And we found that, we published this in the International Journal of STD and AIDS and showed that tuberculosis is one of the most common infections, which have never been described any of those literature before. All those will be PCP, Kaposi's sarcoma. That really moved us, something entirely been different. What the patients, what we are seeing is been very different, because TB is very common in the general population in India, in Africa, and many developing countries. So this made us to understand what is HIV disease in Indian patients, how we should manage. Unless we know this, we will not be able to manage them. We need to suspect what these patients have got, we can't do A to Z all the investigations on these patients unless you know this. Again, we repeated this using the same database after 10 years when we published on a very large series on 6,800 patients. We again continue to see TB is a major opportunistic infection. So TB is a major disease in people with HIV. The next step, what we went into this, some of the biologists, and if you have been working in infectious disease, you know that in such HIV is someone who developed, they are not going to develop the symptoms immediately. It may take years and years. It may take anything from 5 to 15 years for developing various symptoms, as shown by this blue color curve, what is called CD4 curve. The pink one is a virus curve. The CD4 drops down over a period of time. They start developing various infections like herpes zoster, TB, fungal infections in the mouth, pneumocystis pneumonia, and many neurological opportunistic infections like cryptococcal meningitis, toxoplasmosis, and CME. These are all developed when the CD4 is really low. So we try to map when they develop this by doing the CD4 count in our patients, those times. I think this research has been carried out almost like 25 years back. So this is very important, especially for clinicians trying to understand when to target, whether one can have these particular various opportunistic infection, target the various diagnostic tests, and as well as think about various prevention strategies for all these. For example, we have a TB preventive therapy, we have a pneumocystis preventive therapy, simple drugs, and all those we mapped and publishes. Also, we studied whether these infections are using these observational database, they're causing a disease progression. So what we found is there is someone who got tuberculosis in HIV as compared to people who do not have TB, they have 3.5 times faster progression to death. This means very clearly shows that TB is a killer disease in people living with HIV. Pneumocystis is almost 4.5 times, and cryptococcal fungal infections almost 7 times faster progression to death. Then again, the carry-home message is for clinician is look for these infections in this and diagnose them and cure them. Otherwise, you lose a patient because of this. And for guideline makers, so following this type of data, which is published from our cohort here from Chennai, There are groups from Sub-Saharan Africa, population from Thailand and Latin America. So WHO sat together and developed a guideline, what is called AHD, for advanced HIV disease. Hey, patient comes to you at 200 CD4, what he should do, even in a rural setting. Looking at the guideline, you can say, do this test, look for this infection. You can give prophylaxis. You can have TB prophylaxis therapy. You can give cryptococcal meningitis prophylaxis therapy. Also, you can give cotrimoxazole prophylaxis for various toxo and pneumocystis and variety of other infections, what you should look for. So I think this type of research is all very important, not only for HIV, for many, many, many other emerging diseases, which will again be useful to develop guidelines, especially for resource-limited setting for the clinicians, and particularly in the rural health centers. They may not be in the tertiary care center with a lot of facilities to diagnose. Also, we cautioned that it's not just treating this opportunistic infection, we also have to provide antiviral therapy to prevent immunosuppression, to further prevent various opportunistic infection. Following this, WHO made guidelines like rapid ART, don't wait for longer time, start treating the opportunistic, within a week start therapy, or if they don't have any illness, start the same day. So again, having said this, there are a variety of antiretroviral drugs that have been developed. You can see this list. You can some of those French restaurant menu with various types of these medications. And also, some of you do not know about this is act at various levels, starting from viral entry, at the time of replication using various enzymes, at the time of maturations. And very recently, there's a drug being developed called capsid inhibitor called lenacapavir. So these drugs are been wonderful. It's all started used in the US and Europe and in North America, developed world, and the patients were doing well and death rates started coming down. But it's been prohibitively expensive, couldn't be used in our patients. So that's a time the Indian generic companies started developing partnership with the innovator and with the WHO, start developing these in our own backyard. Many of those molecules I'm showing here have been started developing from 1996. When it was developed, it was a lot cheaper. It's like 1/50th of the price of the innovator price. And we started using this in our own patient to save their lives. So when it was so cheaper, everybody started questioning, hey, these generic drugs from India is so cheap, is really safe, is it been tolerable? This type of questions started coming way back in the year 97 and 98. No one have the perfect answer. That forced us, I'm using the word forced us to do a study here in Chennai at our center using the Indian generic antiviral drug and compared with the proprietary drug and stating that, all very safe and tolerable and effective in increasing the CD4 cell counts as compared to the proprietary drugs which are being innovators. And following this, these antiretroviral drugs made way to many, many countries, to Africa, many Thailand, neighboring countries, and now even to some of the research settings where they didn't have a patent issues. So this is something I think this type of research is also very important, which we can do it at our own settings to, take care of very large number of people who don't have access to these drugs. Again, when we did this, many of those national programs, they are not providing free medication. They're all people who have to pay for the medication because there was very little evidence whether this really prevents a death rate in people living with HIV. Reports were there from the US and Europe, there are little. That's a time we used the same, our observational data, where we used this medication, showed that the death rate came down year by year over a period of time. And following this, one of the first programs in a resource-limited setting start providing free ARVs on a large scale was our national program, NACO. And today I feel proud to that more than 1.8 million people are receiving free antiretroviral drugs. And many of those drugs, which are developed in India, is being now moved to Africa and many other resource-limited settings. And now we did a little research and found that 90% of the patients who are living with HIV, they are receiving generic antiviral drugs which have been manufactured in India. You get simple research, short safe, moved into the programs, and now very large number of people are being benefited. Now again, these are all based on the research, which not been sophisticated clinical trials, but are all simple observational studies which are made propelled to very large program. Again, don't sing the glory with this type of research. So this type of research, whatever I've shown, of observation, is can have a lot of biases in capturing the data because it may not be sometimes uniform. Some patient might have certain investigation done, some of them may not be available. And even different doctors can prescribe different types of therapies to your patients. So it can have a lot of biases. So the observational data will have some limited value, but on the other hand, it can have essential value out of that. But if you want to really prove whether this therapy is better than that or not, you need to do what is called randomized clinical trials. So at that time, we had several research questions, in particular in the field of HIV on antiretroviral therapy, especially when you have to start this antiretroviral treatment. I said to you all, this is someone who will develop HIV. It takes years and years for the development of symptoms. So whether you want to start immediately soon after the diagnosis, or you need to wait till they develop any symptoms, or just before they're dying. No one had a perfect answer. We conducted some trials called HPT No Fit and Start. I'll walk you through some of those. These are all randomized trials done multi-site, to change public health guidelines. What to start with? I said about 18 different medications, like a French Western menu. So we do not know what will be the best drug in your setting in India or a developing country. What's the best drug in America? This again can be proved only by doing certain clinical trials. And again, if they fail, when to switch therapy, whether they are failure and what drug to be switched to. This all can be proved only by doing certain randomized clinical trials. So some of the trials we conducted, some of them I'll walk you through soon and show that how we get impacted the guideline. So today, people living with HIV, they live longer. They're like any other normal individuals. But on the other hand, over a period of time, they also develop various non-communicable diseases like diabetes and hypertension. Again, to prevent that, certain trials we did that I'll walk you through. And also there are newer antivirals are being developed. Are they really safe in different populations? It might be safer in Americans because they're different immunological makeup. It might be safer in Europeans. Is it really safer in the Asian ethnic race, and particularly Indian race, and Northern Indian, Southern Indian? Unless you do certain region-specific genetic studies, you'll not be able to know about their pharmacogenomics. Again, you need randomized trials to understand this. And of course, how we can cure HIV. That's something we're not going to touch today on that. To start with, one of those, a landmark trial, which we conducted, how we did it really impacted many treatment guidelines and public policy, a trial called HPT 052, which is a trial funded by the US NIH, one of the largest trials ever funded in the history of medicine, if we put all diseases together, is what is called, can this antiretroviral treatment prevent sexual transmission of HIV, whether it can prevent transmission from one partner to their another uninfected partner, through sexual route. Such a large trial. Also, we want to answer from the trial, but that will also help to understand whether you want to start treatment, a little earlier at a higher CD4 cell count. So when we started this trial, antiretroviral therapy have been provided based on the guideline. Only these people, either with a CD4 less than 200 or with less than 350. That was a guideline, that was the evidence. So this particular trial, what we did is, we recruited people who got more than 350 CD4, who don't need treatment. At that time, there was not part of standard of care, but the CD4 is less than 500. And we recruited all those individuals in this seeding group. We divided into two different groups, randomized trial. One group, we put them on treatment, on antiretroviral drug, which they don't need a treatment, but we put them on treatment, again, with all those study principles of informed consent, getting all those, going through all the ethical reviews and all those approvals and, recruited. Another one group, we waited up to 250 or up to till 350 because certain regions, they started treatment at 250, turned into the 350 because that was a standard of care. They start developing symptoms and they started on treatment or they have been delayed till that period. So again, these individuals were followed over a period of time, also not only on clinical follow-up, but also a lot of safe sex counseling and all the other STIs have been managed, mainly to understand whether this antiretroviral therapy, by reducing viral load, by reducing transmission. Quite a large trial, went nearly up to 10 years. So what we found in that particular trial is you don't even need a statistician. Patients who delayed treatment, there are more chance of transmission through their uninfected partner. So here again, when I mentioned about this partner, they're all serodiscordant couples. So they're all one partner positive, another negative in their married relationship. It was recruited large in India, in our site in Chennai, in Pune, and as well as sites in Thailand, in sub-Saharan Africa, as well as several sites in Latin America, and as well as in the US. So very clearly shows that in this particular trial, by giving antiretroviral treatment, by reducing this viral load, reduces the transmission. And following this got published and been picked up as one of the major breakthrough in the year 2011 and in the field of medicine. Just by treating someone, you can prevent this disease transmission. So following this, the terminology also came what is called U is equal to U. So someone who has got undetectable viral load, they won't even transmission to others. It's a major public health importance. By doing this randomized trial, we were able to show. Now, earlier, it was a huge question. What happened to these people with the epidemic? Will they spread infection to others? will there be a risk to our healthcare individuals while you're doing surgical procedure or anything? Now it's very clearly shows they'll not be transmitting infection to others. Also, certain sub-analysis we did in this trial, we found that it also prevent various clinical events, particularly tuberculosis, at a higher CD4 cell count. So following this, all the guidelines started recommending someone whose CD4 is more than 350, less than 500, to start on this treatment. So the antiretroviral therapy moved up. Is one for preventing sexual transmission, two for good for the individual preventing various opportunistic infection. Again, while we did this and found this answer, we had a question, hey, should we give for everybody more than 500 CD4 cell count? We didn't have a perfect answer at that time. We constructed a very large clinical randomized trial with the support from US NIH and different sites across the globe, and on 4,600 individuals who did not have any symptoms, but they are HIV positive, their CD4 more than 500, randomized one group to immediately start on treatment, another one group we deferred up to 350 because that was a guideline. The study again went up to almost like 12 years across the globe, and again, one of the largest followed-up trial in a history of medicine. And again, going to the results, we found that patients who are on deferred therapy, more chance of developing more and more opportunistic infections, as well as non-AIDS complications like malignancies, diabetes, and hypertension. So that means starting treatment early, which has been a lot of benefit to the individual, even at the CD4 is a lot higher. Now, particularly it prevented TB, lymphomas, and variety of other illnesses. So following these two pivotal landmark trials, both of them, I had an opportunity to participate as an investigator, and as well as our site at VHS recruited very large number of participants for this trial funded by NIH. Today, we completely changed all guidelines. Anyone who is HIV positive, irrespective of the CD4, put them on antiretroviral treatment because it's good for the individual as well as for the public health impact on preventing transmission. Following this, all guidelines way back in the year 2016 to 17, start everyone on treatment. Earlier, we had guideline to start less than 200, 350, 500, completely moved. So then there are certain modeling statisticians. They use this model of putting everyone on treatment because we had a prevention effect as well as preventing various opportunistic infection and transmission. They modeled and they found that it can avert around 21 million AIDS-related deaths and will prevent 28 million new infection by 2030. So that means doing these two landmark trials, how it had impacted globally by preventing disease which caused death, and as well as preventing new transmissions. This is something what's the beauty of this randomized clinical trial, making a huge impact on the public health setting as well as in the treatment practice. The moving away to what the therapy to be initiated, I said about this large group of medications which been developed for HIV disease. So when we started our clinical practice almost like around 25 to 30 years back, we always used to follow the what's been prescribed in the US because there was only data, or in the Europe to do that. They used to give a drug called sidovudine, which used to be the most common drug along with, we use a 3-drug therapy. Significant number of our patients, when you put them on therapy, they'll become very anemic, and we have to disturb therapy, or you have to put them on the hospital, put a lot of transmission. So that's a time, this is almost like 23 years back, I developed this protocol, large multi-site protocol with my other collaborators from the US and Africa. So whether it should be studied against a couple of other regimens. One of those regimens we wanted to study or a drug called tenofovir-containing therapy, along with emtricitabine and efavirenz, which is an older drug as a single pill, versus the therapy which was widely used. I will not go into the very details. So there's a large multi-site studies done in India, Thailand, Sub-Saharan Africa, Latin America, as well as a lot of parts of the US, because US also very interested because they were seeing a lot of adverse effects with their own medications. Why not we study this? And we found that, so this newer therapy, at that time newer, not anymore newer, is tenofovir-containing therapy, which was been non-inferior to the drug which was earlier been using sidovudine. But what they found is the sidovudine-containing therapy, as shown by this darker color, have got variety of side effects, the order of metabolic complications, patients are disrupting therapy, anemia, and lactic acidosis, so many things, particularly in women, following all those guidelines, not only from resource-limited settings like guidelines from India to WHO guidelines to the US guideline. Everyone changed from AZT-containing therapy to tenofovir-containing therapy following this trial called PEARL study. So in this regimen, what we used was tenofovir, emtricitabine, and a drug called efavirenz. And many Indian companies made it as a single pill, and following the robust scale-up happened, and significant number of patients are put on therapy, and they all did well. So what's been found in that is patients put on these efavirenz-containing therapy over a period of decades, it's been reported that they are either committing suicide or attempting suicide. No one knows that. Again, it cannot be found in a randomized trial. So what we did was we went back to those clinical trials, what we did using this efavirenz drug, where there are a lot of case report forms, also captured a lot of behavioral data, especially about someone's behavior, in that what proportion have reported about their suicidality or anything. And one trial which has been carried out in India was called A5175. We found in these studies, all retrospectively analyzed, found that there is a 2.6-fold higher risk of attempted or completed suicide to efavirenz. So this is such a dangerous, did very well in the clinical trial, showing efficacious, but we put on implementation programs where we found that they have been reporting on this. And following this now, this drug has been moved out of our guidelines. And there is another drug called dolutegravir, called integrase inhibitor, which was been studied, again, in a randomized clinical trials, which was compared against efavirenz and found very robust by showing this, where they have a higher superior virological efficacy over a period of years. And also they found that in comparing to the earlier efavirenz drug, there's called less side effects. The yellow one was with efavirenz, the other one was a newer therapy, dolutegravir, and became part of the guideline from developed world. So at that time, there was no evidence in a developing country. That again made us to do a study quickly in India at our center here in Chennai on a 560-odd patients, and a 6-month follow-up saying that it's been very efficacious and tolerable, gave evidence to WHO and many guideline makers. And today, every guidelines are moved to this particular drug combination of tenofovir, lemivir, and dolutegravir as a preferred therapy. So, and made a reality. So again, so this has become the first line therapy. So again, patients need not do well all those years throughout them. If they tend to miss the pills, it can fail. Then we may have to switch from one line of therapy, second line of therapy. So for that also, we conducted different trials, and over a period of time, different second line regimens were there. This is something very recently we conducted trial on patients who have been failing first line therapy. We randomized to 3 different groups, and with a large sample size calculation. And we found very interestingly, there are regimens which were not recommended earlier became superior as compared to the standard of care as shown by the different guidelines. For example, 2 NRTIs and booster darunavir, which was been earlier recommended, or any PI, which has been a little non-inferior as compared to recycling the same failed drug called tenofovir to second line, along with the dolutegravir if it was not used, or a newer regimen of dolutegravir and darunavir. So following this, we are expecting a new guideline change anytime soon based on this pivotal study called DEP study. You can recycle one of the drugs with another powerful drug, and also the newer regimen. This is something what randomized clinical trials move the guidelines from one regimen to another. So following these now, WHO guidelines and other national guidelines are moving from one regimen to another for a better therapy in people living with HIV. So there are better modeling research had been done. This is again a good research work which has been published a couple of years back. The modeling side is based on these different regimens in first line, second line therapy on the longevity of the patients. And they found that in this modeling study, they found that someone who is HIV positive, the viral load is suppressed and good CD4, their life expectancy is similar to persons who don't have HIV. So that means today, HIV disease is yet another chronic manageable disease. They live like a normal people. If they take this therapy, they have access to treatment. These are all possible based on the signs on the RCTs, randomized trial, and we and others have done, and based on that, these different therapies are being available. Again, so these are all randomized trials, which help to change these guidelines for the various therapies. Once these therapies are being introduced, following the guidelines, programs and clinicians start using this, the next thing what we do is what is called implementation science studies. You implement all these, then you find out, are they really doing well or developing side effects? So one such study, which our group and others have found in that is a drug called tenofovir, which I said earlier in the trials, on using on a longer duration can produce a chronic renal disease, renal dysfunction. It's been seen anything from 2 to 6% of the individuals over a period of time can develop this. So following this, the guidelines very strictly recommended that using this drug, you have to use a creatinine monitoring for the renal function test. And if they are being altered, you need to look at other alternative drugs. So this is something all being developed based on the implementation science studies, based on implementing these guidelines in your settings. And also the manufacturers of this drug, tenofovir, that the Gilead, what they did is they went back to their organic chemists who developed this molecule. They developed a pro drug of this tenofovir. In a way, they found that if you do it at a lower dose, and give at the stage of prodrug, you can reduce side effects, and they develop this what is called tenofovir alafenamide. So now it has been approved for use, and now it is becoming part of the guidelines in many research settings using developing countries in the process of using. This is again following the implementation science. The manufacturers have developed this molecule, now tested, now it's becoming part of the guidelines. Also, there are researchers found that you put patients on this therapy, they do well, but on the other hand, based on these various implementation science, they can develop a molecule. Can you drop this molecule, which has been offending drug, if they have been doing well, so that they'll continue to do well? So there is a study being carried out called Gemini study. What they did is they compared this triple therapy, tenofovir, emtricitabine, dolutegravir, on people who got suppressed viral load. They dropped to tenofovir. They continued only the two drug therapy of dolutegravir 3AC, and they still found that they continue to do well, the same virological suppression. And following these, US FDA and other regulators have given approval for this particular two drug combination and recommended to suppress viral load to prevent kidney and various other toxicities like bone toxicities. You can change them to this. Now, it is not part of the developing country guideline yet. We are looking forward to get this anytime soon because we have got enough evidence on this. Hopefully, WHO guidelines will move forward with this new guidance anytime soon. So this is, again, the beauty of doing RCT, again, to prevent various morbidities and mortalities, which is causing by these various medications to individuals, even while doing goodness to them on suppressing the virus, but it can cause toxicities by doing this. Again, whatever you do this, patients have to take this medication. So if we take for example, of with this virus, they have to take the drugs every day. Then only you can suppress the virus, maintain the suppression. But what happens is our individuals tend to miss pills. Of course, we all miss pills even for an antibiotic. And when it start missing pills, they can develop a resistance. To combat this, now we clinicians and researchers are working closely with many nanobiologists. Can you develop certain long-acting, instead of taking every day, can you develop as an injectable or a depot or a subcutaneous or pill, whether which can be taken every 2 months or every 6 months? Yes, the answer, now there is a molecule being developed by the nanobiologists as a nano preparation of these two antiviral drugs, which has been tested in different clinical trials called LATE2 and many other studies. We'll walk about that one soon based on the time available. And where if you give this every 2 months instead of patients taking every day, which they tend to forget and cause resistance, which has been very comparable. Just one trial I'm showing this, just walk up time. So what they did was they compared everyday oral therapy versus every 2 months versus every 1 month of injection. What they found in that is every 2 months of injection, they got a higher virological suppression, as shown by this bar chart of 94% as compared to 84%, which is oral therapy. And also, in this particular trial, they also have various questionnaires to find the satisfaction of the patient, because not everyone like to have an injection. It may harm them. Sometimes they'll say, oh, why should I go there for 2 injections? So they found that 88% of the study participants who are receiving intramuscular injections, they're very satisfied as compared to only 43% oral tablets. So that means it has been very acceptable by the patients. So this is also research, not only randomized trials, but also in this randomized trial, we also construct various other questionnaires to carry out what is called preferred user characteristic. When you're going to use this, are they really acceptable to them? Also in this particular study, they also found small proportions that we not give every 6 months injections. Now there is another new drug called, the nacapavir has been developed for every 6 months. These are all been developed based on this research been carried out, what the need for these study participants, and based on that all this been developed. Again, having said about all these different medications, today we are able to make people live like a normal people. But on the other hand, what's been seen based on our own research here in Chennai and others, what we have done in other countries is our patients continue to be morbid, not because of any opportunistic infection, but for various non-communicable diseases like diabetes, hypertension, cancers, and other things. What causes this? Initially, we didn't know, hey, people with HIV at 35-year-old, they develop a diabetic or heart attacks or various kidney disease or osteoporosis. We found that because of this viral disease, there are a lot of inflammation. COVID, we found a lot of inflammation in HIV, which causes accelerate these various non-communicable diseases, as well as HIV, various opportunistic infections and microbial translocation, obesity, all these causes various inflammation causing this. Again, unless you cure them, you'll not be able to prevent this. Right now, we don't have any cure remedies, but we can only suppress the virus to undetectable level. So to prevent these various non-communicable diseases, one thing we picked up were cardiovascular disease, which is very common in most of the settings. So we did a very large clinical trial, large multi-site trial, which is done in India, in Thailand, again, Africa, US, and Europe, and many places funded by NIH and ANRS in France. So what we did is people with HIV who got undetectable viral load were doing well. We randomized them into two different groups who never had any cardiovascular disease, who had got a normal lipid profile. One group, along with their antiretroviral therapy, they were randomized to statins with pitavastatin, another group just placebo. And this trial went to 9 years of follow-up with various clinical follow-up of looking into cardiovascular events. In a nutshell, I'll just show you, if you see on the left panel of your slide, what we found in that is patients who received statin, almost 35% reduction of any major adverse cardiovascular events as compared to placebo. So that's a huge finding seen in this particular trial called REPRIV. And also, if you see on the panel on the right side, there is 21% reduction in death in people with HIV due to any cardiovascular disease as compared to placebo. That means this statin, which are not needed for this individual because their lipid profile is normal and they didn't have any cardiovascular events, by putting them on that, it prevents various these things. Initially, we didn't know that. We clinicians, we always prescribed statins in people who have got abnormal lipid profile, particularly LDL. Here, they all have a normal LDL. We had no idea why this caused this. So we went back from the stored sample from these various trials where some of the participants are given informed consent to use a stored sample for future use. We did various proteomic analysis, particularly looking into several proteins. What we found in that is the statin causes increase in the expression of a protein called precols protein, which causes actually the prevention of athermatous plaque deposition, we call NCP. So what happened in that is there was a doubling of the precols expression in patients who received statin as compared to placebo, which causes huge reduction in this athermatous plaque deposition, which in fact causes reduction in cardiovascular events. In fact, what decreased lower density protein did not correlate with any changes. So that means we might be rewriting the cardiology textbook related to statin, that statin have a lot of other effects, especially on suppressing inflammation and producing the expression of the protein and doing different aspects in preventing various diseases. And following this, there was a big news report everywhere on this particular landmark trial. REPRIV got published in New England Journal of Medicine. Again, our hospital, VHS, our clinical site at Infectious Disease Medical Center, contributed to the maximum number of study participants for these global clinical trials on 7,000 odd patients. And following this, now, not only the developed world guideline, a lot of developed world guideline like US guideline, British guideline, European guideline, all recommended now someone who is HIV positive more than 40 years, statin is recommended irrespective of their cardiovascular status. And now we are seriously discussing with the WHO experts and guideline makers how we will implement this, because we need to feed the whole world with these statins because of these now 40 million people living with HIV. Hopefully, this will become a guidelines anytime soon. Again, this type of RCT can make a huge change in preventing some morbidities that never occurred in people with HIV. Now, over a period of time, when they live longer because of our science, which make them to live longer, but on the other hand, they can develop these non-communicable diseases, which can be prevented. I'm going to conclude my statement talk stating that, so research what we did here at VHS at our hospital at Infectious Disease Medical Center have contributed significantly. We have not just published 400 odd publication, but described the clinical spectrum and natural history of HIV disease from a developing country. We studied the safety and tolerability, efficacy, and effectiveness of Indian-made generic drug. It was a cheaper, not usable. Now it has made a history across the globe. And we documented the impact of this antiretroviral therapy, reducing death and opportunistic infection, studied the various adverse effects, short and long term, conducted various types of RCTs, helped to change global guidelines, and as well as also calling for various other research questions for the future researchers. Many of them being contributed to WHO guidelines development. I think something is very important. When you do, how you impact the guidelines and prescription practice, large number of clinicians to use this. I'd like to conclude my talk stating that, in summary, there's an urgent need for clinical research and clinical trials in all fields of medicine. So we need in diabetic research, we need cardiovascular, we need in variety of infectious disease. Now we are also developing an antiviral for dengue. So right now there is no viral drug for dengue. Someone develop a dengue, you just give a paracetamol and IV fluid. Significant number of them comorbid. Now we have been developing molecules, and doing clinical trials on that. And again, to do this, you need to learn how to develop a research protocol. Identify a good research question. And some of you would have heard in the earlier talk in the last month on about how to develop a research protocol. It's very important. And also you also need to learn where to conduct a clinical trial, what are the infrastructure doing that. That itself can be a lecture, how to conduct all these various trial. And also, you can't just get people and put them on drug. It will be highly unethical. It has to go through various regulatory bodies like our own institutional ethics committees, IRBs, and also our national bodies like Drug Control General of India. If you're using any international money, you need to go through our health ministry screening. All these things you need to learn if you are conducting good multi-site international clinical trials. And very importantly, what to do with the data. Not just presentations and publications, but also make sure that how we can impact your own guidelines. Also, we use this data not only just for the clinicians and the medical practitioners, but also use this science to develop various public policy for the non-medical people, how they can use this in their own setting in preventing a disease or taking care of themselves. So with this, I'll end my talk and happy to take any questions. These are my collaborators across the globe and also various funders starting from NIH to ICMR to industry, WHO and United. Without their help, we would not have done all these studies and whatever, I have shown. Thank you again very much and happy to take any questions depending upon whatever time I have. Thank you again.
Thank you, Dr. Kumarasamy. That was quite exhaustive, amazing, and truly inspiring. The amount of work you've done over the last 25 years, it's truly stunning. So, if I can ask the first question, where would someone start for the clinical trial? For someone in the city like us, if I want a clinical trial, I'm going to come to you, right? So that connect is there. But if someone who's in a 2-tier, 3-tier city and really interested in research and clinical trial, now how would they start? What would they start? What is your advice for them?
Yep. Good question, Dr. Beg. So I think when I said about clinical research, you're not just trials alone. There are a lot of things. You can do epidemiological research. You can do observational studies and variety of research. So to do such research, you don't need a very larger pot of money or funding or anything. You don't need any sophisticated setup. Only thing you need to identify a research question, what you're going to research, and you can start collecting data by developing all those appropriate ethical approvals and collecting data. These are all simple clinical research, but these can have biases. So these biases can be prevented only in doing randomized clinical trials. The trials which I said, like phase 1, phase 2, phase 3, if you're going to conduct, they can be conducted only in certain institutions or certain hospitals where you need to have the appropriate facilities. One, you need to have the appropriate patient population. 2, you need to have the appropriate trained investigators. Not all medical doctors and clinicians are good researchers. And also not all good researchers are physicians. So you need to have a balance between both. You can be a clinician and can be a good researcher. You don't need to be a good clinician, but on the other hand, you need to have the proper medical training, but you can be a researcher. So I think you need to have the appropriate person, because doing a clinical trial is not just giving medication. How you marshal your crowd of your researchers, your study nurses, your data management team, your QC team, how you move forward to collect ethical data and do the data management, do the quality assurance, and do all this. You need to have the appropriate individual who lead the show. And to do that, you also need to have an appropriate institution, which have got all the facilities. So you have a data management facility. They have an ethics committee, which is most important. Without an ethics committee and institutional review board, you can't do any clinical trial. And that also, not only having an ethics committee, but they also should have a requirement, and they also should be accredited by your national body. Today, all the ethics committee should be accredited by the Drug Control General of India as well as by ICMR before you do any trials, because they look into the minor aspects of all those details about who are the ethics members, what are those SOP they have. And also to do certain type of randomized trials, you also need a good laboratory backup. So the labs should have the appropriate assays to do variety of assays, equipments. They should be calibrated. They also should have an appropriate certification like NABL, and as well as accredited to all those various quality assurance agencies. I think to your question, Dr. Vivek, I think unless you have all those set up, particularly in tertiary care institutions, it's very hard to do a good randomized clinical trial.
But collaboration is the key, right? So if
I think you can have brilliant research questions. If your institution don't have a setup, you can collaborate with an institution where they have all the facilities to do that. Again, there are facilities you can have, but unless you have a research funding, you'll not be able to do. So again, you need to have a good protocol, approach a funding agency, to get the appropriate resources.
Dr. Rewari is online too. I'm going to invite him to share a few comments. Dr. Rewari has done some phenomenal work in this field. So I'm going to, with your permission, I'm going to ask his comments also.
Thank you, Vivek, for organizing this very important session. And Kumar, it was a very exceptional, beautiful presentation, well illustrated with all the studies of which you have been part of these studies, many of these studies. And it was very clear and concise to begin with. What is clinical research? What are other types of research? And basically phase 1, 2, 3, 4 trials, which many people are not very clear. So you have provided all the evidence which has gone into WHO guidelines. From time to time, they have been changing. And as we know that after any new drug comes in, a very important part you have mentioned is phase 4 trial, or what we call as pharmacovigilance also. And based on this pharmacovigilance, we know many of the drugs, particularly antiretroviral drugs like stavudine, nevirapine, these have been phased out, particularly stavudine, because of its side effects. So that is all very important part of any clinical research, that phase 4 trial and pharmacovigilance is a part of it. Also, you have mentioned a lot about the clinical trials, the drugs. You also mentioned in the beginning about prevention trials. A very important prevention trial which has demonstrated U equal to U. That is undemonstrable is untransmittable. And this also came from trial, particularly the initial trial HPTN 052 and then partner 2 study. So these trials provided evidence that with the viral load suppression undetectable, you can prevent the sexual transmission. Moving a little bit away from HIV, another trial which I came recently, very recently, it is yet to be published. We know that for hepatitis C, we are using SOF-VELPA combination, and SOF-VELPA combination is given for 12 weeks. So I came across a study which has just been done and submitted for approval, but the data from 700 patients shows that SOF-VELPA for 8 weeks may be as good as for 12 weeks. So this is going to make a very big change. And if you remember, recently in the EASL, another guideline was released, which showed, and it was work by Amit Goel from SGPGI, which showed that entecavir 0.5 milligram may be as good as 1 milligram, simplifying the treatment. So these kind of trials basically different are very useful in simplifying treatment and also preventing the side effects. So thank you very much for excellent presentation, Kumar.
Thank you, Dr. Rewari, for your kind words. I know when you are on the other receiving end in the program, I know that how you are scientific using these published clinical trial data and move the public health in the national program. I think that's always been recognized.
So we have a couple of questions. The first is one from Dr. Jayashree Joshi. Would you advise screening of all individuals undergoing any blood test, HIV test or CDR counts?
No, we don't recommend. It is not a recommendation, but it's currently happening. If you see it in India, so you go to any hospital, get admitted, they'll give a laundry list of tests. So one of the things is HIV before surgical procedures all been done. But there is a national, we have a regulation. So who need to be tested, and if you're doing an HIV test, what are those various procedures you need to do? You need to always do a counseling on the individual. Also get a consent before you do. Because that. Someone who is HIV positive, they can, it can be a stigma for them, as well as a variety of consequences they go through. So I think to this question, not everybody should not be tested by law. And if you want to test for certain indications or for purpose, you need to always get a consent. Earlier we need to have a written consent, but now that is all waived. Now at least a oral consent. You need to do that before you do a test. And if they're positive, you need to appropriately either do the appropriate counseling and link them to the treatment, or refer to a center which does all these, because now they have treatment available.
The next question is from Dr. Abhishek Bharadwaj. Any trial going on for HIV research by Ayush treatment with modern medicine?
So I do not know, but there are a variety of other trials going on in alternative fields of medicine. I do not know whether it's happening in Ayush. So, but we don't see anything had shown anything significant to recommend as part of the clinical practice. But there are alternate clinical trials being, going on.
One Dr. Vijay Anandraj is asking.
Just to add to that, Kumar, basically there have been trials in the past, particularly with the Siddha medicine in Tamram, and Alka Deshpande also did some trials with Ayush medicines, but they were very small sample size and they were not very conclusive to show that there is benefit. So as of now, there are no studies which have shown any benefit with any of the Ayush medicines. Over.
Yep.
Next question, I think it's a lecture by itself, but maybe you could be short. What are the key considerations for managing HIV in pregnant women and children?
So right now in pregnancy, we don't do anything different. I said about for the trials, anyone who is HIV positive, irrespective of the CD4, start them on treatment. So good thing that happened is someone who's pregnant, the chance of transmission of HIV to their newborn could be up to around 30%, before the antiretroviral therapy era. Now with this potent antiretroviral treatment, because of the viral suppression, the chance of transmission of TB is almost to less than 2%, almost 0%. In fact, in our hospital here in VHS, we do deliveries almost every week. In fact, we had a delivery even yesterday here. I've never seen a new HIV positive newborn, the last 18 years or something, which is almost a big record here in our hospital here in VHS. So there is nothing different we do. We just treat the mother, and if the viral load is suppressed, they will not transmit infection to their newborn.
Again, Vijay is asking us how emerging technologies such as AI or wearable devices impact clinical trial design and conduct.
So AI is used in everywhere, so only thing we need to really validate, because AIs are being developed by our other counterpart engineers, could be computer science engineers or mathematicians. I think this is time that they have to work very closely with the clinical people on the validity of many of those AI algorithms in predicting a diagnosis or narrowing down the treatment options and so on and so forth. And having said that, how it can help to design a protocol, definitely yes. And AI we have been using for quite some time, right? When you have been developing a protocol, what are those drugs could be useful? We use a word called meta-analysis. Now, using these tools, using AI tools, we can further narrow down. I think to your question, AI will be helpful, but on the other hand, how far helpful, how useful as compared to them, we need much more larger studies. I think it's very important. Everybody talk about artificial intelligence who can replace the medical doctor, but that's all not true, at this time. So we need a lot of other validation of these and uses at the particular settings.
Gabriel Rodriguez is asking, of course, I think one thing which I'm sure I can answer is HIV screening mandatory for all surgical procedures? And as a surgeon, I'm sure it's a must. I think everyone should be screened. But his question is counseling a must to do pre-op HIV test?
So I said about the HIV test, I think any test you do on anybody, you have to get their counseling on them. Hey, I'm going to do an MRI scan on you. Is it okay? You can't just like that force someone to do that. But in HIV, it is mandated by law, Indian law. Maybe Dr. Rewari can correct because he worked closely with the lawmakers that informed consent is needed before you do a testing. Dr. Rewari, do you want to correct that?
Yeah, absolutely. There is a HIV Act which has been passed by the Parliament, and you cannot test a person without the consent. You can offer a test if it is called provider-initiated testing and counseling, PITC, whereby a provider feels that this person requires test, then they can offer the test, but there is always a opt-out option. So the right lies with the patient to take a test or to refuse the test.
Yep, thanks.
Thank you, sir. And one question is, what are the common reasons as to why a clinical trial would fail?
So, not all the molecules which are developed, which are tested in lab experiments will make it to clinical practice. So many times when we test in the lab, by take an organism, a bacteria, you put a newer antibiotic, it can kill in the lab. So need not work in a human being, where in the real life settings. So you can work in the lab, Then based on that, it is developed into a phase 1 trial in a healthy volunteer, whether it is tolerable and effective. Then you test them in a smaller group of people to phase 2. It can work, or it need not work. It can get killed because in a real life situation, because a drug to work, variety of factors. Somebody have to swallow or injectable or one other way. It has to bind to our own mechanisms, either to the plasma proteins. It has to metabolize. Then the effect of this drug has to that particular organism, either to kill or inactivate or whatever thing. It can happen easily in a laboratory, but need not happen in a human laboratory. It need not happen. There are various other factors. So because of these various factors, not all molecules which are developed from lab will go into phase 3. I told about experience about on various trials where even we have participated and I was been involved in doing a trial where, and all put into that, I did a small analysis and found that only 25 to 30% of the molecules which are developed, which has gone through phase 1, phase 2 will go to phase 3. Remaining all will be killed. So that means, to your question, very large number of drugs, which are molecules which are developed variety of disease, will not make it. And again, in the phase 3, again, drugs can fail. Need not fail. Sometimes it can show inferior to the current standard of care. So that means why we should change the current standard of care. We always in a phase 3 test against the standard of care.
Okay. So I think there's one more. The last question was on by Sri Ganesh Venkatesan, who's again asked on AI-driven software solutions. I think which you've answered that. I think with that, we come to the end of an amazing session. Thank you, Dr. Kumarasamy, once again for this phenomenal session. Thank you, Dr. Rewari, for commenting and being with us through the entire session. For all the participants, there's a link which has been provided in the chat box. So please fill up that. Give us some inputs. Give us some ideas as to what you want to hear from us. And of course, only if you fill it up, then we might be able to generate a certificate for you, a certificate of participation. So thank you once again, Dr. Kumarasamy, and thank you, Dr. Rewari, and everyone else who's participated. And hope to see you soon again. Thank you.
Thank you.
Thank you so much.
Thank you.
Thank you.
Close.